First thing, Julia, you may have noticed I crossed out some questions. I don't like nitpicky trivia memorizing. If we're going to memorize something, I'd like it to be at least be important and useful. And you already know I don't like memorizing stuff to begin with. So number 2 to me was really, I don't care about that. Number 1, the function of the nucleolus is, what is the nucleolus? Uh-oh. Yeah. Like that's a good start.
It's a little ball inside of the...
Oh, okay. So what does it do? Makes ribosomes? I think they're pronounced ribosomes, but I'm not a biologist, so I'll accept ribosomes. By the way, if the answer was to protect the nucleus, what are we talking about? What protects the nucleus? Okay, there's a nuclear membrane, so I would have crossed that out. What protects the chromosomes? That I'm not sure of, but I don't think it's the nucleolus. To make DNA, that was that whole procedure. We did a little jigsaw activity where I gave you pieces, and there was messenger RNA, and then there was all sorts of stuff there. So I don't think it's that. It's definitely C. Sorry, I can see that I've creeped you out there, Halia. My bad. Now you're happier, right? You can explain that joke to Adele later. Okay. Which of the following statements is not true? A gene stores the information to make a particular protein. I think that's true. Usually for questions like this, if I think an answer is right, I just put a little check mark next to it. If I think an answer is wrong, I'll put a little X next to it. And then I'll go back, and if there's only one X, that's what I go with. If I got 2, I got to read those 2 more carefully. All the genes within the nucleus will be copied to make protein at some time. I don't think so. I think some of the genes are turned off, aren't active. The liver cells have genes that aren't turned off. The stomach cells have different genes that aren't active. The brain cells have different genes that are not active. So I'm looking at B, different cells. That is true. All of your cells have identical DNA, just some of them are turned off through... By the way, when I say turned off, how are they turned off? It's a chemical process. You can dive into that and study your PhD in that if you really want to. Different proteins have a different sequence of bases. That's correct. Yeah, B. The function of the Golgi body is... What does the Golgi body do? Hang on, we got somebody here who's late. Answer A? Sorry, what did we say? Number 4, answer A? Is the answer A? Okay. Which of the following steps for the production of a protein is incorrect? So this is that little jigsaw puzzle activity that we did. The DNA message is copied into RNA. Now, it did say incorrect. I don't like multiple choices that ask you to pick the wrong answer. I'd rather pick the right answer, but okay. I think A is correct. The nucleus receives a chemical signal. I think B is correct. The manufactured... Oh, you know what? D is incorrect. DNA never leaves the nucleus. RNA, half of a copy of a DNA strand, is what leaves the nucleus and goes out into the cytoplasm and the goop to make the protein. So D is incorrect. If it had said mRNA, messenger RNA, or just RNA, that would be correct. Chromatin is a substance that contains several molecules of DNA, a substance within the nucleus that contains DNA and proteins, a substance that unfolds before cell division. Actually, chromatin forms during cell division, so I know C is wrong. A segment of DNA that codes for a particular protein. That's wrong. What that phrase, a segment of DNA that codes for a particular protein, that's a definition. What's that a definition of? That's a gene. I'll make even a little note for my studying here. That's a gene. So it's A or B. A substance within the nucleus that... I think B. I'm going with B. B. I'm not wild about that question. To me, Brianna, that's too vague between A and B. I tend to try and make my stuff clearer. A gene mutation is... a change in the specific order of the... Okay? A change... Yeah, B is wrong. C is wrong. D is right. Yeah, A. Okay. I nuked number 8, yes? On your copy too? Good. Cystic fibrosis is an example. That's definitely a negative mutation because it tends to shorten lifespan. And I think the white coat of the spirit bear would be a neutral mutation, I think. Doesn't increase or decrease their lifespan or chances of reproducing or anything like that. Okay, some matching. Whenever I see matching, I always 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Dope. If there was 6 and 6, I can always use a process of elimination for the last one. Mitochondria. Mitochondria. Sorry? I think D. Changes sugar into... They're the powerhouse of the cell. Chloroplast. That's C. That's photosynthesis. Nucleus. F? Sound good? Nucleolus. You know what? First of all, let's cross out the answers that I've used. F. Not sure. I'll come back to it. Golgi body. I think that's A. Right? Endoplasmic reticulum. I think that's B. And I think... Well, I know G, surrounds the cell membrane, is not the nucleolus. So, is it D, C, F, E, A, B? Yeah? Good. I think I'm going to ask you something similar to number 17. Number 17 says, explain how the nucleus controls the activities within a cell. How would I mark that? I would say something like this. Proteins determine what a body cell will become. That's the first. I would talk about proteins, I think. The nucleus contains DNA, which contains instructions on how to make protein. That's a long sentence. I don't like that. Here's what I would say. If you want the short version, Jordana, nucleus contains DNA, which contains protein formation instructions, and proteins control everything else. I think that's a shorter version. So how does the nucleus do it? Nucleus contains the DNA contains the instructions to make proteins, and your protein, primarily, with other stuff, but most of you is made up of protein. So a liver cell will have different protein structure than a stomach cell will have a different protein structure than a skin cell. So that top one you could use, that bottom one, I would probably give you some clues, tell you how many marks, and tell you kind of what I'm looking for. Turn the page. First of all, what did we call things that cause gene mutations? Okay, so these are, if you're looking for a vocabulary word, mutagens. Give me one. What? Sorry? Yep. Give me another one. Give me another one. Give me another one. Sorry? I didn't hear. Viruses? Yes. Give me another one. Okay, let's go. Right, toxic chemicals, sure. There's more, but I only asked for 3. I'd probably make it worth 3 marks if I asked for 3. But there's 5. Any more? We did a list, right? Okay. It says construct a flow chart that outlines how a protein is made. Use the terms below and use additional words to connect the terms. You can make a little note here if you want. See page 131. You can stay on your quiz. I'm going to go find page 131. Nope, too far. Nope, not far enough. Page 131. Will I get it on here? Yeah. This page here. Okay. I am not going to ask you to reproduce this whole thing, but I might give you 1 or 2 or 3 steps and say which ones are in the correct order or something like that. So that's quiz number 4. That's for you to hang on to as a study guide. I'm not going to collect it. Chapter 5 practice quiz, this one here. Figured out, Adele? And Sirki, she gets everything right? Okay. 3 stages in order are D, yes? Purpose of DNA replication is to, I think, make an identical copy of the DNA. Is it D again? I'm going from memory. I'm not using my answer key. Correct order of the stages of mitosis: prophase, metaphase, anaphase, oh, geez. It's got to be D. It's the only one that starts with prophase. How do the cells differ, or how are they different in plants and animals? That's wrong. That's wrong. That's wrong. Is it B? The only one that doesn't seem wrong to me is B. I don't know if I'm going to ask you something that detailed. I like 1, 2, and 3. Those are good questions. Which of the following does not describe cancer growth? A. In fact, the definition of cancerous cells, they are uncontrolled growth, right? I'm not going to ask you 6, I don't think. I'm not going to ask you specifically to try and match what type of life forms reproduce in what way. But I think A. No, that's wrong, Mr. Dewick. It's D. Yeast reproduce by budding? I didn't know that. Learned something new. I'm not going to ask you number 7. It was cool math. I think we looked at it. We got a really massive number after about 30 generations, right? But after 8 hours, 2, 4, 6, 8, sorry, 2, 4, 8, 16, 32, 64, 128, 256? Yeah. If you're looking for the short way to do it, holy smokes, exponents are useful. It's 2 to the 8th on your calculator. But I'm not going to ask you one like number 7. Which is a disadvantage of asexual reproduction? Why is that a disadvantage? Yeah, if something goes wrong in the environment, the odds are pretty good that the entire colony is going to get wiped out unless there's a couple of mutations that happen to survive that. And that does happen occasionally. Bacteria and things mutate very, very quickly. What are some advantages? Well, B, C, and D would be advantages. That's a question I like. I think I'll ask you something like that on your test. Which of the following is not a method of vegetative reproduction? I'm not going to ask you that. Spores, just because spores is a different way of asexual reproduction. Which of the following are correct steps in DNA replication? Oh, are they wanted in order? Okay. I think the first thing that has to happen is the DNA has to unwind, which I think I'm going to quit and say D is the correct answer. But if I wanted to go further, so that happens, then once the DNA untwists, it splits in two, enzymes separate it, because now you have two halves, but because A match... Oh, by the way, I'll absolutely ask you what A matches with and what C matches with. That's going to be on your test. So 2 would be next. Then new bases, I think, then 1, then 3, I think. 1, 2, 6, 7... Oh, okay, prophase. I think, is it? I'm not sure. Come back to it. Telophase, that one I know, the 2 nuclei separate, but you still have 1 cell, right? That's definitely... Oh, no. What? Cytokinesis. Completely separate. My bad. This is... I don't like these definitions. Okay, I know metaphase is B. That I got in the middle. And F is a way apart. Interphase, the DNA replicates. I know that. Do the chromosomes dissolve in prophase, or no? The chromosomes form in prophase, and they're still there in telophase. So I'm going to cross out G and say that's wrong. Well, it's according to my answer key, but I don't like that. What I would instead have said for prophase is the cell starts to prepare to divide and chromosomes form. And what I would say for telophase is you now have 2 nuclei inside a single cell. That's what I want you to know. I'm not going to be asking you the stuff about spindle fibers and centromeres and stuff like that. Okay? Turn the page. What are 3 requirements that must be met? Sorry? We're going to be saying... you're right. NDA. DNA must be replicated. What else did you say, kiddo? There must be nutrients so the cell can grow. What did we call these? We call these checkpoints. These are some of the checkpoints that had to occur. And the third one is DNA must not be damaged. These are the things that prevent cancer from happening all the time. Because if cells were allowed to divide in those situations, there's a greater likelihood that something would go wrong in the cell or in the DNA replication, and mutations are generally not a good thing. Some are neutral, a few are positive, but we prefer them not to occur. How are binary fission and budding similar? Okay. Both produce clones, identical offspring. I would say that because they're both one-parent asexual reproduction. Oh, did it just want one thing? One thing, good enough. How are they different? Yeah, sure, that works. Okay. Fission, a single cell makes a copy of itself. Budding, it's usually a multi-cell little mini copy. Disadvantages of asexual reproduction. What are some? Okay. Offspring are genetically, what's the fancy word for genetically, what's the fancy word for genetically identical? Begins with letter C. If you said clones, I'd be fine with that because I tend to agree. Okay. Why is that a disadvantage?
Because a negative mutation can destroy.
Okay, so you know what? I should probably include that because all I've said is this fact. I haven't said why. So negative mutations can destroy a whole colony, and usually we call them colonies. Give me, let's say, 3. 2 more. You're right, loud and proud. So offspring compete with each other for resources, for nutrients, all that stuff. Yep. Okay. One more.
Bad weather.
I'll be even more general. Unfavorable conditions can wipe out the whole colony. And by the way, that's so when you get sick, Jimmy, you with me?
Yeah.
When you get sick, typically you're having some kind of virus or bacteria and it's spreading through you very quickly through binary fission. But as soon as your body starts producing antibodies, that whole population gets wiped out and you get better. So that happens inside of you all the time. Every time you get sick and every time you get better, that's basically what's going on here. Okay. Again, you can hang on to this to study from. I'm not going to collect this. I have a couple of more things for you to work on.